All right, well, thank you everybody for coming to this session today. Really appreciate the interest and hopefully we'll be able to generate a lot of discussion, but please feel free to come find me afterwards. As mentioned today, I'm a postdoctoral fellow currently at the University of British Columbia. However, I'm going to be talking about something a little different and mainly starting in the fall. And so to kick things off, nature is filled with diverse microorganisms and microbial communities that provide us with life-saving therapeutics. And in particular, the soil-dwelling microbes Streptomyces have been a classic source of products that are critical for clinical therapies, surgical procedures, agriculture, and veterinary medicine. Now, the class of antibiotics that I have worked with extensively are the aminoglycosides. These are sugar-based antibiotics, with the majority of these compounds consisting of 6-membered or sometimes 5-membered rings that are linked together. And there's a beautiful level of chemical diversity within this drug class, owing to the hydroxyl and primary amine functional groups that decorate these molecules, the latter of which are really important in terms of their mechanism of action. Now, aminoglycosides target the bacterial ribosome, disrupting the process of protein translation, resulting in a pathogen creating a collection of non-functional proteins that will eventually lead to cell death. Now, as with other classes of antibiotics, pathogens have evolved or acquired means to protect themselves against the activity of aminoglycosides and become resistant to treatment. The most phased form of resistance is through drug modification. Pathogens produce enzymes capable of chemically altering the structure of the aminoglycoside, rendering it unable to bind to its intracellular target. These enzymes can be divided into 3 different groups based on the type of chemical modification that they perform and what functional group on the aminoglycoside that is going to be targeted. While 2 of these 3 subgroups target the hydroxyls, primarily the largest subclass are those that inactivate aminoglycosides by modifying these primary amine functional groups. These enzymes are referred to as N-acetyltransferases, and not only are they the largest subgroup, but are also the most diverse. They are responsible for the small modification of transferring an acetyl group from the molecule acetyl coenzyme A to one of the primary amines on the antibiotic. The diversity of this subgroup is reflected both in their amino acid sequence and structure. To date, we know of 3 structurally distinct protein family folds that are capable of modifying aminoglycosides to confer resistance in the expressing host. And what I've thoroughly enjoyed studying throughout my doctorate is the diversity in their function. The underlying chemical mechanism of how this acetyl group is transferred can be facilitated by these enzymes in several ways, and the resulting resistance phenotype can also be varied. There are several different aminoglycoside inactivating enzymes through acetyltransferases that can inactivate several combinations of drugs. Lastly, the genetic information that encodes for these enzymes has been found in both gram-positive and gram-negative bacteria, varying environments, as well as both chromosomally encoded and found on mobile genetic elements. Now, each of these 4 attributes I like to look at as pieces of a puzzle. Leveraging this knowledge helps us to design less resistance-prone derivatives of aminoglycosides to combat resistance. Now, in 1975, at the Second International Symposium on Antibiotic Resistance, an innovative naming system was produced in order to describe these key attributes, the foundation of which to understand genotypic and phenotypic relationships being the sequence and functional data. This naming convention acts as a code for the different aspects of the enzyme's function and connecting that to a protein sequence. And so to start is a 3-letter abbreviation to describe the function of the enzyme. AAC in this case represents the acetyltransferase. This is followed by a number in brackets that captures the regiospecificity, which is referring to the site on the molecule that is going to be modified. And then following this is a set of Roman numerals, which describes the resistance phenotype of the host that is expressing the enzyme. And these phenotypes are traditionally determined through antibiotic susceptibility testing, where the growth of a pathogen is assessed on an antibiotic-containing medium for survivability. Each Roman numeral is a different code for a different combination of aminoglycosides that are susceptible to modification. So for example, AAC3-Roman numeral 2 can confer resistance towards tobramycin through acetylation, but it cannot for the antibiotic neomycin, but it is able to against gentamicin. And then lastly, to capture the sequence diversity within a subgroup of AACs, unique genes exhibiting identical resistance profiles are differentiated with a lowercase letter. And so together, capturing these relationships is a critical component in the development of stewardship and surveillance programs across sectors. And when we establish a complete picture of the puzzle, this strengthens our ability to develop these programs and allows us to better design next generation aminoglycosides that are less resistance prone. Now, for over 50 years, these AACs and other aminoglycoside modifying enzymes have been studied extensively, with the initial research focusing on the sequence function relationships that the nomenclature was initially designed to describe. It wouldn't be until 1993, the review that I have up on the slide, that they discussed so critically the relationships between the four pieces of the puzzle analogy that I have, characterizing these enzymes in both a clinical and environmental context. Now, unfortunately, this is where the breakdown of the nomenclature begins, starting with the R&D of next-generation aminoglycosides to escape resistance mechanisms that were clinically relevant. Enzymes that were circulating in the environment, agriculture, or veterinary medicine settings were characterized, but they were not the focus of the narrative in the literature. What this caused was a shift in the understanding of what this naming system was designed to describe. Since enzymes and clinical pathogens were all found to be of a single family protein fold, the two red three-dimensional structures I have up on the slide, it was incorrectly assumed that all AACs had adopted this fold. And so this also led to the assumption that the naming system was describing this single family protein fold for all AACs. Now, in the 2000s, there was a lot less interdisciplinary collaboration, which wouldn't be as much of a detriment to R&D if data reporting and sharing practices were consistent. But unfortunately, with the lack of consistency and collaboration, this has compounded the problems growing with the nomenclature. And of the several hundreds of studies that I've curated between the years 1993 and 2017, only the three that I have indicated up on the slide have drawn attention to the structural diversity that likely exists within this AAC subgroup, but yet they don't have crystal structures available to confirm that, or the lack of maintenance of the nomenclature standards. And so the distortion of what was known or unknown has really worsened. And one of the most important pieces of the puzzle to become compromised because of this is the function, where you can kind of equate it to a puzzle piece that is just completely blurred. Not only have the misannotations accumulated, but I've actually found cases in the literature where the arbitrary picking of which aminoglycosides are included for susceptibility testing has led to the potential incorrect designation assigned for the resistance mechanism. Now, this level of disconnect between the puzzle pieces, you could think of as a red herring from a mystery novel. Like detectives, scientists are right now from different disciplines being misled, following up false leads, interpreting clues differently, and this wastes resources for an incorrect or incomplete conclusion to this puzzle. And so, for example, one of these AAC3s Roman numeral 2 could be actually an AAC3 Roman numeral 4, completely different susceptibility profile that you're going to be using to inform a stewardship or surveillance program. Now, in recent years, interdisciplinary approaches to studying these resistance mechanisms has increased greatly, and in part this is due to the increased growth of One Health initiatives, which is recognizing the interconnectedness of human health, animal health, and the health of their shared environments. Now, unfortunately, this doesn't change the fact that the misannotations are growing. Standards for how we perform the susceptibility testing for this antibiotic class is still lacking. And so as a biochemist and microbiologist, identifying these inconsistencies during my PhD, nearing the end, I started to think about how can I do more to facilitate and support interdisciplinary collaboration. I want to not only produce quality data and raise attention to these issues in the literature, but I also want to be a part of setting the standards. I wanted to learn how to curate this type of data. How can I do a better job of reporting my data as a biochemist, microbiologist, and improve data accessibility across disciplines? And so to do this, I proposed a solution to my colleagues at McMaster University who maintain the CARD. The Comprehensive Antibiotic Resistance Database is a rigorously curated collection of characterized peer-reviewed resistance mechanisms and associated antibiotics and contains tools for sequence analysis. The first step to the solution I had in mind was working with CARD to undertake the management and adoption of a single aminoglycoside modifying enzyme nomenclature. Abiding by the guidelines first proposed back in 1975, I worked with their team to restructure their antibiotic resistance ontology for these modifying enzymes in order to reflect the hierarchical nature of the nomenclature. These guidelines will be laid out on the website as part of what I call ARCS, which is the aminoglycoside resistance classification system. And so breaking down each component of the nomenclature like building blocks, I'm going to be sticking with the example of the acetyltransferases to walk through the basics of the current ontology structure. With the first part, the parent term is the phenotype. So again, that Roman numeral representing the aminoglycosides that are susceptible to modification. Reiterating what I had discussed earlier, incomplete panels for susceptibility testing procedures produces inaccurate nomenclature assignments. And so each parent term right now in the card is connected to a complete catalog of susceptible antibiotics with a confers resistance to antibiotic relation term. This then connects to the regiospecificity as the AIMR card gene family. And again, the regiospecificity is the site on the molecule where the acetylation is going to be happening. The different regiospecificities for the AAC class are then all connected to the functionality group, aminoglycoside of acetyltransferases, with each described under this parent term for acetylation of antibiotic conferring resistance. Now, in addition to developing this structure and reorganizing the antibiotic resistance ontology within CARD, I wanted to produce a means to visualize these ontological relationships. And so what I aim to build with the CARD team is a new way to interact with this knowledge. As you can tell from my presentation so far, I'm very much a visual learner. And so I like to leverage my neurodivergences to identify patterns, how other minds might not be able to do so. And to help bridge this gap between different disciplines and provide support for the assignment of identifiers for these modifying enzymes, I want to consider different learning styles and means to digest knowledge. And so similar to the schematic that I've shown with you today, I want to build some type of an interactive interface to help people from different disciplines better digest this nomenclature, which is a very brilliant naming system, but can be really hard to understand if you're just reading about it in text. And so I'm excited to officially tackle all of this when I begin a joint postdoctoral position with research groups at Simon Fraser University and McMaster University in September. And with the data that I have and continue to curate, I will start by leading the retirement of alternative nomenclature systems, updating the current CARD entries to adhere to the nomenclature guidelines, with alternative designations retained as synonyms. So we still have that ability to connect the different studies in the literature. Now, the final piece to this puzzle, I wanted to develop a means to guide susceptibility testing, building it in a way that is accessible, again, across disciplines, facilitating interdisciplinary collaborations, and providing a means for researchers to assign a name to a newly identified enzyme variant. This tool I incorporated in the title of my talk, AIIM, with a play on the Roman numerals that are used to represent the resistance phenotypes. And this stands for the aminoglycoside inactivation identifier manager. AIIM will utilize protein sequence, susceptibility testing, and the option to submit biochemical data to assign an identifier to a modifying enzyme following the standards that will be set by the ARCS. Common to existing tools, AIIM will first assess the similarity in a protein sequence to the library of aminoglycoside modifying enzymes in CARD. The distinguishing feature of this online tool is its capability to evaluate if the combination of aminoglycosides selected for susceptibility testing meets the minimum requirement for an accurate naming designation. Users will then have the option to provide biochemical data that supports the enzyme function in order to improve the tool's ability to distinguish between one or more possible classifications. Now, this is an oversimplified summary of the data that I've curated from the literature to date. Right now, it consists of 31 structurally unique aminoglycoside antibiotics, all of the different resistance phenotypes verified with susceptibility testing or biochemical analysis or a spectroscopic characterization of the inactivated molecule. But I've provided a short summary to kind of show you how the inner workings of the tool will be making these decisions. And so the first example, just to show you the difference between the phenotypes of Roman numeral 2 and 3, if you have a highly similar protein sequence for an aminoglycoside and acetyltransferase with the regiospecificity at position 3, distinguishing features in your susceptibility testing would be the antibiotic kanamycin. Sometimes those antibiotics, it's not enough, and that's where determining the regiospecificity, where on the molecule that modification is happening, is going to be needed. And so this will allow us to recommend namespace for these modifying enzymes prior to publication, and also suggest additional analyses to resolve any conflicts. So again, really trying to solve that problem of preventing people from cherry picking what antibiotics are going to be in the susceptibility testing and relying on sequence similarity to tell them what the name of the enzyme should be. And of course, this can't be done just us at CARD. It's really going to be a huge community effort. And so prior to deciding to pursue this project as a full-time commitment, I guess, I've presented this work and have talked to many different colleagues in the aminoglycoside research space. And there is definitely the wide agreement that such a tool is needed and such management of the nomenclature needs to be improved. And so I'm really excited to really dive into this starting in September, and really happy to be here for the next few days to discuss with all of you how we can better incorporate this tool. If you have databases with similar data being stored, how can we make sure that we're harmonizing the nomenclature and really taking the pieces of that puzzle and making them clear once again to have a consistent story that we can share across sectors. And so with that, a few acknowledgements. Firstly, to the Bio-Ontologies track and the ISMB organizing committee for inviting me to share this research here with you today. And they also provided me with a travel fellowship to help cover the costs of joining you at the conference, which I really appreciate. Dr. Andrew MacArthur, who's believed in this concept and really kind of helped me bring it to life since I had the idea nearing the end of my PhD. And my partner in crime, kind of like my detective partner, Sherlock Holmes and Watson, Brian Alcock, who's the lead biocurator at the CARD. And then the two photos, my husband, who is my financial funder for attending this conference, apart from the ISCB Travel Fellowship. So he is at home watching the live stream right now. And so appreciate you so much for helping to send me to Montreal to share my amazing work that I love. And then a photo of my father, who passed away during my PhD. And so it means a lot to be able to present this in Montreal because this is where he grew up. And this is my first talk since post-pandemic. So thank you so much for listening, and now happy for questions.
Thank you, and I'm sorry about your father. One thing that leads to divergence of nomenclature is lack of systematicity, right?
Yeah.
And I'm wondering whether you've thought about how the ontology that you're proposing connects into other ontologies that are capturing, similar information, but perhaps not for these specific
antibiotic resistance.
Yeah. So can you talk a little bit about how you might kind of connect your ontology into other kind of more general ontologies with broader coverage?
That definitely is something we're starting to work on more actively with the CARD to kind of like abide more with, I believe, OBO Foundry, like traditional ontology infrastructures. And so specifically with the aminoglycoside resistance elements, it's something that we're definitely still actively working on and want to make sure working with other databases to really harmonize how they're processing that tool or the information from our tools. And so a really great example that I really want to be aware of is glycan data. I attended a biocuration conference a year ago and went to a glycan functions workshop. And these sugar-based antibiotics, these enzymes that are inactivating them to confer resistance are highly, highly similar, if not identical in some ways, to the ones that are involved in cell wall biosynthesis of bacteria. And the information that the two spaces are producing are almost identical, but we have no means to exchange that information and learn the chemistries from each other, which was another inspiration behind developing this tool, and not just for antibiotic resistance, but similar chemistry in small molecules as well. So
yeah, I think it's really important. Otherwise, we'll end up with more bifurcation,
No, and I mean anything to save the nomenclature, because right now, if you were to look on NCBI for an AAC3 Like you actually really don't know if that Roman numeral designation is what it actually is. So,
Thank you.
yep.
So thank you very much for this talk. Very, very interesting work. Now, it reminds me a bit when the gene ontology was developed. The reason it was developed was that knowledge about protein functions evolved, and we needed a way, people needed a way at some time to annotate to evolving knowledge.
Yeah.
It seems to me that so that they can annotate at different levels and use the hierarchy. Now, it seems that a lot of the inconsistency in the nomenclature that you describe arise because you insist, or this field seems to insist, to always describe these antibiotics at the most specific level.
Yeah.
But even when not enough phenotyping or not enough knowledge about this structure is yet known. Is there a way to use this ontology to solve this by not forcing or not allowing people to use this nomenclature, basically describe all levels, if not all of this is already clarified, but basically use the ontology structure to annotate or to use a term that is a more higher level term, essentially, than the most specific one that you would eventually want to end up with?
Yeah, and so, I mean, we have like over 50 years of these names floating around out there, right? Which is why I wanted to stick to this hierarchical nomenclature, and also the power in capturing that functional data, because it's not just the antibiotics that it confers resistance to, but where on the molecule, which is really important for when you're designing derivatives to combat resistance, as well as sequence information with that lowercase identifier at the end of a name. However, that's not to say that people who are going to be like doing the research can't call it, you know, something different. And for surveillance purposes, what can be harmonized across sectors is the assignment of these identifiers, right? And I think if I'm understanding your question correctly, the overarching, like those AACs, like that 3-letter abbreviation at the beginning tells you most of what you would need to know on a broad strokes level. That function of this enzyme is conferring resistance to aminoglycoside antibiotics through this means. I mean, I'd love to chat with you more about it after, because I feel like there's a lot of different layers to your question, which gets my brain rolling, but yeah.
Thank you very much.
I think we're at time. Yeah?
Yeah. I actually have a question.
Okay.
Yeah, I'll ask a really quick question.
No worries, yeah.
Very cool work. I love language and names, and it's really, really important that you get it correct. So I'm curious your thoughts on how you want to backdate that problem, right? So you said 50 years, right, of naming
Yep.
problems. And so how, for the next students that want to search for this or other things, how do you go
Yeah.
through that process?
Oh my gosh, when I look back at how I like started this, it was, you know, I'm an autistic ADHDer. And so for the longest time during the beginning of my PhD, I was like, am I just not understanding something when I'm reading these papers? Because it's calling the enzyme this, and like, that is not the function, right? And I thought it was a me problem. And so I started making an Excel sheet, like literally hundreds and hundreds of papers and all of the susceptibility data. And I was like, I am not wrong. Like, this is a problem. And literally, I mean, now we have the machine learning tools to like help us going through the literature, but I did all of this manually. Just every time I read a paper, I was adding to the table, adding variables to that table, going through it. And now I have this, my husband's also an amazing programmer, so he's helped me actually build the database from my Excel spreadsheet of all of these relationships. And so it's really just paying attention to the history. I did bring this here, so if anyone's interested, this is the actual book of the proceedings from that 1975 conference. I was able to find it on Amazon, and is the most brilliant piece of history and fascinating read. And so for anybody who needs to kind of like start this in their field, go back and read where it started, right? Stop listening to like the word of mouth throughout the literature of like, oh, this was done and there's a citation for it. Like really go back to the beginning and have that appreciation, because that's how we're going to fix these problems.